We all have a story to tell. Let's tell yours. Welcome to the Intellectual People Podcast with your host, Jason. Come together and listen to journey stories and more from interesting people. Welcome your host, Jason.
Today on the Intellectual People Podcast, I have Dr. Robert Malone. How are you doing today?
I'm doing great. How about yourself?
Very well, thank you. Robert, please tell us what you invented.
Oh, that's an open-ended many things. The topic that has everybody's focus right now has to do with the core technology platform that has given rise 30 years later to these mRNA vaccines that has the world enthralled. But I have many patents, and I have a long history in published peer-reviewed literature of innovating in the area of vaccine development and vaccine-related technology and gene delivery-related technology, particularly non-viral gene delivery. So I've really been at the forefront of that for the first third of my career. And that's where the inventions have occurred.
How did you get involved in medicine?
That's a good question. These things go back to childhood experiences, of course. That's the usual storyline that folks give for that question. In my case, both my father and my father-in-law were deep in defense industry, and they're both engineers. Both were mentors. Both made major contributions to the defense industry. My father-in-law ran Raytheon Special Projects Division, which is basically their spook shop. And he went back to the avionics for the SR-71. And beyond that, in World War II, he was right at the forefront of the development of radar technology. My dad developed high energy systems, was what he was focused on. And that has to do with low inductance capacitors that are very hardened that they use for the bunker bombs, and exploding bridge wire and exploding foil, which is the technology that's currently used for triggering thermonuclear devices because it's very precise. So I came from that background of being the son of engineers, for better or worse, and the son also of a teacher.
Okay.
English teacher, as you can, my language skills, and they both, my mother in particular, wanted me to go into medicine. I rebelled against that in my youth, and for a while I was a farmhand and a carpenter and those kinds of things, growing up in the Central Coast of California, in Santa Barbara area, and then I went back to school and became very embedded in computer science. This is 1980 to 1982. Graduated from a community college in Santa Barbara with a associate degree in computer science and multiple certificates, best computer science student and that kind of stuff. But I had just gotten tired of working in a basement, looking at a monitor and having my interactions just digital with the system and the software and the programming code and everything else. And so I decided, well, I did want to go into this hot new area of biotechnology called molecular biology, and didn't want to spend the rest of my life sitting in front of a computer screen in a room with no windows. So I like to say that now I still sit in front of a computer screen all day long in a room, often with no windows, but I make about a third of what I had made if I had stayed on the computer track that I'd started in the early 1980s. We still had some punch card stuff that we did as part of the training back then. So just to give you some time frame.
Sure.
My dad actually built a kit computer back, in those days. So I do have this kind of span, and the computer science taught me logic. in a really rigorous way. I think it's one of the things that makes me different from a lot of my fellow physicians, is having had that logic experience. So I went to UC Davis. I transferred to UC Davis. Couldn't get into Berkeley. So there it is.
Sure.
But I had this farming background, and that fit with the logic of UC Davis. And I was married. My wife and I were high school sweethearts. We kind of eloped to the mountains below Lake Tahoe for a while before I went back to school. So we were very much a couple. And so returned to UC Davis and just became passionately embedded in the development of technology relating to medicine. And in particular, my mother was afraid of breast cancer. She was just frightfully afraid of breast cancer. And I had an opportunity, as a new third-year student at Davis, I had the opportunity to do rotations as an undergraduate. And so I found this advertisement for a pathology lab that was working on breast cancer, that was doing molecular virology. And that's kind of was the pivot point. I went to work for a senior pathologist named Robert Cardiff. He actually wrote a letter of recommendation to me recently for the big prize committee. I was told I needed to put a letter in. So he's been a long-standing mentor. He's a very severe gentleman, notorious for that. I remember my interview with him, and I was trying to be deferential and circumspect, and he turned to me, looked straight in the eye, and he said, "I have no time for false modesty." And I was like, "Whoa, okay, you're no longer in Kansas anymore, Robert." But he took me on, and it was he had just come off of sabbatical with the Bishop and Varmus lab, who had received the Nobel Prize for oncogenes. And the chairman of the department at the time was a character, really a true character, named Murray Gardner, who had been the founder of the USC Cancer Center and had been recruited to run the Department of Pathology at Davis by enticing him. Bob Cardiff told me this story the other day. I'd never known it before. They were trying to recruit Murray to head up the department, and they didn't have enough money to do it. And so they asked him, what is it that he really wanted? And he said, Alice, his wife, really wants a red barn and a pond. and so they scurried about, and they found some rural property around UC Davis that had a red barn and a pond. And by God, that did the trick. So Murray was really a character. He had been mentored early on after, he came back from World War II. He played an early role in the war on cancer, and was busy doing crazy things down both first on the East Coast and then at USC, where they would do stuff like isolate viruses from chicken tumors and try to isolate them from tree tumors and all kinds of things. Wow. So I entered this laboratory environment naively with just driven passion to learn molecular biology and to get into the mainstream of this new technology space. And at the time medical school was an impossible dream. It was so insanely competitive back then to get in anywhere. And so I was what they call a bent arrow. I wasn't a straight arrow, going, fast track for everything. And so I thought my prospects were poor, but if I became a good biochemist and molecular biologist, well, that would be an okay fallback if I didn't get in. And so that was the thinking, was kind of risk mitigation in education. And I just grabbed onto it and became passionate in the laboratory, spent every moment I could outside of my coursework, just incredibly long hours, both in the lab and around learning molecular biology and biochemistry, which at UC Davis was a tough road. I mean, it's not chemical engineering, but it's just a notch below it. So this led me, the breast cancer work was with mouse mammary tumor virus, which is a retrovirus model of breast cancer in mice.
Okay.
That allowed one to ask questions about oncogenes and eventually tumor suppressor genes, et cetera. And I became the worker bee to do a lot of the heavy lifting in terms of isolating DNA and RNA from a variety of different cell types and tumors, and working with this wild mouse colony that didn't get breast cancer very frequently because it didn't have any native mouse mammary tumor virus, which is very odd. And so I just learned the craft, and it was at a time frame when sequencing, dideoxy sequencing, which much of the technology had also been developed at UC Davis, strangely, was getting off the ground. And northern blots and southern blots were the main stem of that technology space at the time. That's all obsolete now. Everybody just sequences things or does PCR. And I had to make everything. I had to make the radioactive probes. I had to do the whole kit caboodle, and became quite good at it. I was a little bit OCD. I guess I still am. And I did really well in the lab. And in a way, I learned more in that laboratory over the next 2 years, both about science politics and about working with nucleic acids than I did ever since. It really created the foundation. And they just let me at it. They treated me like I was a graduate student. And so a series of things happened in that lab. It's just being in the right place at the right time, which I guess is kind of my story overall, is being in the right place at the right time, or my wife says I should say chance favors the prepared mind. But the Davis lab, the Davis Pathology Lab was affiliated with the California Regional Primate Research Center. And the research center had this colony of non-human primates that had an immunodeficiency syndrome that Murray recognized as being similar to the immunodeficiency syndrome being observed in male homosexuals, particularly in San Francisco, which is just down the road from Davis. And so it turned out that Preston Marks and Murray and others in the lab, including the electron microscopist played a key role, made the first observations of retroviruses being involved in an immunodeficiency syndrome in a non-human primate. It was published in Lancet. And that was all happening around me. I was getting rigorously grilled on a weekly basis about my, the positive controls and negative controls and what's the hypothesis, and blah, 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 blah. And meanwhile, this thing happened. And at the time, I remember still interviewing for my MD, acceptance into medical school. It was absolutely known. As a lesson in medicine and groupthink. Everybody knew that AIDS was caused by semen getting in the blood and the abuse of amyl nitrate. And so I would walk into these interviews saying, no, it's a retrovirus. And you're crazy. So that's the way that goes. So Murray and Preston had this observation with others and found this retrovirus in the macaque tissue. And then there was this guy at the Pasteur Institute that said he had found the actual virus that was causing it in humans. His name was Luc Montagnier. And if you've ever seen the HBO video when the band played on, there's a series of scenes where Bob Gallo and others travel from the US to visit the Pasteur and visit Luc Montagnier. Well, Murray was one of the guys on that trip. And Murray came back, and I'll never forget, he almost dancing down the hall, coming back into the laboratory saying, I have the virus that causes AIDS in my pocket. So he brought it through customs, with no paperwork or anything,
Right.
in a Eppendorf tube, and that was how the lab got that reagent, and then that set in play a whole cascade of things having to do with Bob Gallo and hardball politics. Murray tried to set up a large BSL-3, so high containment laboratory for doing the AIDS research in the monkeys. And the local community got in a big uproar over that and the risks associated with it. And Bob Gallo, I clearly remember Bob Gallo called Murray and threatened him that he would never get another grant unless he stopped working with the French virus and started working with the American virus, which is the one that actually, it turns out, Bob Gallo had snitched from Luc Montagnier. That's another long story. So I got to see hardball politics front and center. People think that science, and I used to think that science was this elegant exchange between intellectuals, as we're trying to have now, right. And it's all about the truth and honesty and integrity. No, it's about money and power, and at the top level. For folks right. like us, you and me, that are more kind of in the middle tier, it's not. It's a different thing. But at the top tier, it is about money and power and fame and immortality and all that stuff. So I came out of this intensive opportunity in a small sheltered laboratory in a little bit of a backwater environment, UC Davis, but it's technically at the front edge.
Right.
And I was accepted into an MD PhD program with a full scholarship at Northwestern in Chicago, which my wife will never forgive me for. We were both from Santa Barbara, and we had no idea what Chicago winters meant. And so after 2 years of that, she was like, I can't stand this anymore. We got to get out of Dodge. And as they say, a happy wife is a happy life, and the converse is also true. I applied for graduate school, and I was taken in. I really wanted to do gene therapy using retroviruses. I thought that was the thing that would allow me to use my knowledge base and passion for retroviruses and understanding of retroviral biology. And this is what I thought, that the gene therapy, the concept of Ted Friedman, that gene therapy using retroviruses was going to go mainstream. And there would be gene therapists in every hospital. And I thought, okay, well, I could be at the forefront of this new specialty. Cool. And so that was the strategy. And, and reeling back, I had gone up this risk mitigated triage thing, and then suddenly found myself with a full scholarship for an MD PhD, which I never would have expected back then.
Right.
So here we were. And Jill was not happy after 2 years in Chicago. And I did my GREs, and I just knocked it out of the park, which is not surprising after, you've come out of this pressure cooker and in terms of my education at UC Davis, and then 2 years of medical school. So my scores were basically off the chart. So I could pretty much go wherever I wanted for graduate school. And UC San Diego had Ted Friedman and Inder Verma at the Salk Institute, who were two of the leading retroviral gene therapists in the world. Ted Friedman has actually come up with the whole idea of gene therapy. So that was the ticket. I was going to go there, and I was going to bring, really become a hardcore virologist, and particularly with this application in mind. And so we traveled down to San Diego in an old Cadillac. That was a trip, quite literally, and with a baby at the time, our first son, that had been born at the beginning of my really at the end of my first year of medical school. And so down to San Diego, did my initial rotations, and Ted Friedman told me that he would not take me into his laboratory to work on retroviral gene therapy, largely because he thought it was a dead field. And it had been frustrating and things hadn't worked out for him. And he said that what I could do was come on, if I wanted to build an ordered cosmid library of one of the human chromosomes, which I'd done enough sequencing to know that was going to be just like watching the paint dry. It was going to just be deadly. And that's another one of those fork in the road where I probably blew it, because if I had taken that opportunity, I would have been at the forefront of the Human Genome Project. You know, it's dumb. But this is the way life is.
That's right.
You pick your choices, and it happens. So Ted Friedman wouldn't take me in unless I worked on basically the forefront of the genomics project. And I was really strongly warned by some faculty at UC San Diego, which is right across the street from the Salk. So the Salk is on the cliffs of La Jolla and on Torrey Pines Road, and UCSD main campus is just across the street. And then all of the big, what's now become a biotech giant hub, further down Torrey Pines Road towards the golf course, most of that didn't exist. There was a couple companies, and there was Scripps. So I was really warned not to go to Inder for a variety of reasons, some of which were just kind of only sideways alluded to. It was known to be an insanely competitive, cutthroat postdoctoral laboratory where postdocs, in these big postdoc labs these days, one of the strategies is to put multiple postdoctoral students on the same project and make them compete against each other within the same laboratory, which creates dynamics that are just not healthy.
I'm sure.
People stealing each other's data and reagents and sneaking in at night, and everybody gets paranoid, and it's just crazy.
That is crazy.
And so I went to this. Inder took me in first as a rotation student, and then as a graduate student. And I was the only graduate student in a postdoc lab of about 18 or 19 people. The senior postdoc, Dinko Valerio, went on to create a company called Crucell. And Crucell got sold to J&J, and that's the basis of the J&J vaccine. So this all kind of threads, you'll see this threads back historically to this time when I was in Inder's lab and at UCSD between 87 and 89. So that's what kind of my origin story is in terms of bringing us up to the point where the discoveries started to happen. So I've set the stage and why did I go into medicine and what was my passion and the vision and all that kind of stuff.
So if you can, Robert, what was the process of the mRNA basis? How did that come about in the lab in 1986 to 1988, somewhere in that time frame?
87 to 89.
87 to 89.
And so it was a series of stacked discoveries that probably couldn't have happened in any other place. It was very much a series of events where I encountered key technologies and applied them to suit my needs and my focus. And it loops back a little bit to Davis with Murray Gardner and Preston Marks and the non-human primate discovery and the early days of AIDS. The initial focus was on vaccines and developing a vaccine, and that was Murray's passion for good reason, and that of Preston and the Primate Center. And so I had already been steeped in vaccine technology. And I'd done a rotation as a graduate, during the summertime at Northwestern with Bob Lamb, who is an influenza virologist of considerable note. So I was familiar with influenza molecular biology and vaccinology, and then also very familiar with the forefront of vaccine development with the AIDS agent, let's say LAV, HTVO3, HIV. And so I landed in this laboratory, and what one has to do as a graduate student is come up with something that is both obscure enough and significant enough that you're going to have space to develop your thing and some original knowledge, but you're not just going to get bowled over by the big boys that are going to compete with you. So you know, you have to thread the lock a little bit. And one of the big issues with the retroviral vectors was how they are assembled and packaged. And in particular, what's called the packaging sequence, which is at the front end of the retroviral RNA. Just to recap, what makes a retrovirus retro is not that it was discovered in the 60s, but rather that the central dogma of biology is that DNA makes RNA and RNA makes protein. So it goes in one direction: DNA, RNA, protein.
Okay.
And retrovirus is one example of an agent which refutes that dominant paradigm. It was discovered that retroviruses, although they're RNA viruses, they exist in the genome of those that it infects, including us, as DNA. Well, that's a paradox. How does that happen? It's as a virus particle, it's RNA, so it's transmitted as an RNA virus, and yet it becomes DNA somehow and is found in the genome of the animals that it infects. So that problem was solved by various people, including a guy named David Baltimore, for which he received the Nobel Prize. The gentleman that worked as a postdoc in David Baltimore's lab to characterize that enzyme, called reverse transcriptase, that makes RNA back into DNA, so it's reverse transcriptase, was a young Indian that had come from one of the top Israeli PhD programs named Inder Verma. And so Inder was the guy that I was going to work for. So he was not just a gene therapy guy. He was one of the top people in the world in control of gene expression and in the fundamentals of retroviruses. So one of the key problems was that there were these cell lines that had proteins from a retrovirus that would complement a defect in a retrovirus genome. So that's how this system works. The same thing basically happens with the adenoviral vectors, like for the Johnson & Johnson vaccine. So gene therapy technology fundamentals, in the case of the retros, the molecule that gets packaged, assembled as a retrovirus, is an RNA, and there is a specific part of that RNA that directs the machinery of the other retrovirus proteins to package it into a final virus particle. And that had been defined vaguely, but not precisely. And information about how that worked was not really known. So I thought, and it involved RNA structure and folding, complicated stuff that's still hard to solve. And so that's what I decided to work on, was how does RNA get packaged and what are the specific sequences in the packaging sequence domain that are responsible for that.
And how is that done, Robert?
So that it had never been done.
Right.
And so to get there, I had to build some fundamental technology to allow me to ask the questions.
Okay.
And so that I set about to try to work on how to do this process. And the logic was simplistic and naive. There are RNA viruses like polio that you can purify polio virus and put it on cells and, just scratch the cells, do almost nothing. And it will, a little bit of that RNA will get into the virus, into the cell, and it'll produce live poliovirus. Okay, so poliovirus RNA is infectious. Retrovirus RNA is not. So the question was, can you get enough retrovirus RNA into a cell, a packaging cell that has all of the proteins necessary to assemble a retrovirus, and cause that to yield virus particles that would be infectious?
Yeah.
So in order to do this, I had to develop a system for efficiently delivering RNA, and there were none that were very efficient at all. And so that required that I build a system that would allow me to test new methods and develop new methods for delivering RNA into cells. So the system that I built was based on a reporter gene that had just, this is a gene that's used a genetic sequence that produces a protein that can be readily assayed in a sensitive linear assay. That's the ideal, okay? And the technology that everyone was using at the time was called chloramphenicol acetyltransferase, and it required a radioactive tagged reaction followed by thin layer chromatography. So this is biochemistry talk, but the bottom line was it was really kludgy. You could, people published on a single assay. You'd have a single assay and some controls. There would be no statistics. There would be no solid quantitation. But this is how things were done with chloramphenicol acetyltransferase. And it was radioactive and messy. And, it's just a horrible thing.
Sure.
And, it would take 2 to 3 days because you'd have to expose a film to it. And I had just finished a rotation before I went into Inder's lab with this guy, Suresh Subramani, who had developed and just published a whole new reporter gene system. And it involved the protein that makes the firefly glow, which yields photons in its reaction. And so, as an engineer, photomultiplier tube technology is pretty cool, and it's highly sensitive, and you can detect single photons with PMTs with great sensitivity. And so this protein that makes the firefly tail glow has the somewhat unfortunate name of luciferase. It has nothing to do with the devil. I always have to say that for certain cohorts that might be listening in. It's just the name that they chose for the firefly protein. But this was cloned, and Suresh and his team had shown that this could be used as a reporter gene. And there was a company at San Diego that had developed the first luminometers, which is the tool that's used to detect these photons very sensitively and characterize that. And so I just happened to have access to what was then emerging new technology that was much, much more sensitive than chloramphenicol acetyltransferase, and cost pennies an assay, was not radioactive, and I could run 100 samples if I wanted. Certainly run everything in triplicate or quadruplicate and get statistics and all that good stuff. And it was linear over 4 logs, so just an awesome assay. And and you can convert it back to total protein. So I had luciferase as my core, that I could put luciferase in as mRNA into a cell, and that cell would produce the protein, and then I could pop that cell open, add the substrate, and it would emit light, and I could quantitate exactly how much protein was in there within minutes, as opposed to multiple days. So that was cool. That was a core part of the technology. And then I was working with this protein and trying to develop this assay, and I had an opportunity to, I was Tony Hunter, who's a brilliant scientist and Nobel Prize also ran at the Salk. At the time I was there at the Salk, there was at least 6 Nobel laureates.
Wow.
One of them was Francis Crick, nice guy, by the way. And Jonas Salk was still there. So it was kind of a high-powered think tank on the cliffs in La Jolla. And of course, Jonas was a vaccinologist. It was March of Dimes money that built the place. So really a temple to science and vaccinology. And Tony Hunter was there and just has a steel trap of a mind for new ideas and new technology and new findings. At the time, we really didn't have the internet. I mean, you couldn't go on PubMed and search the literature, but Tony was basically PubMed in his brain, and he knew all kinds of stuff that was coming up. And so he told me that I should get in touch with this young faculty member at UCSF named Judy White. And so I went to Judy White's lab, and they had some, they weren't working with luciferase at the time. They were working with chloramphenicol acetyltransferase. But they had a greatly improved RNA cassette that had structural elements that you would put before your open reading frame, that would be luciferase, and after your open reading frame. And they were working with these RNAs with reticulocyte lysates. So these are a type of blood cell that you can extract to get proteins that would make other proteins from RNA. I don't think they were working with Xenopus, but they were asking some of these fundamental questions. And from them, I learned and obtained a plasmid. This is a circular piece of DNA that had these key RNA sequences that were necessary to get efficient translation, production from RNA into protein of transcribed, in vitro transcribed, that means, synthesized in the test tube RNAs. So I brought that back, swapped out the luciferase for the chloramphenicol acetyltransferase, and then built, nobody had really done large-scale preparations of RNA at that time. They would make very small quantities, and the enzymes were really expensive. But I needed to have large quantities of RNA in order to ensure that I could do reproducible experiments with different variables using the same initial preparation. So I scaled this up to yield milligrams of RNA, which had not really been done before. And so I built the core expression plasmid off of what I'd obtained from Judy White's lab, developed the system for producing the RNA at high concentration and high purity in a stable fashion, and then started working with this. I never did get the transfer of the, I spent just months on building the retroviral mRNA construct. And I was able to get a little bit of signal of virus particles coming out that could be detected in a plaque assay, but it really just didn't happen. And Inder was very disappointed in me. And he did things like, once he brought me a stack of other people's grant applications and said, read these and get ideas, Robert, which ethically is just, a little mind bending. But that's, that was the ethics there. Again, being, big lab, a lot of the rules get bent. So he was disappointed with me. And, but this core method that I developed, Tony came to me one day and he had another one of these ahas. I know some information for you, Robert, young Robert.
Yeah.
Listen to this. I've got something for you that will solve your pain. And it was a paper. The paper hadn't even come out yet. And he told me to talk to a guy named Gordon Ringgold at a company called Syntex in the Bay Area that had this new discovery that you could take positively charged lipids. We can talk science here. So they're actually quaternary amine based lipids, and mix them with negative polynucleotides. So RNA and DNA are basically a linear polyanionic molecule. And these would spontaneously assemble. This had been, I'd worked with all kinds of different liposome preparations trying to deliver the RNA. And the entrapment of polynucleotides inside a liposome is extremely inefficient and time-consuming and expensive. And you're lucky to get an entrapment efficiency of 10 or 15%, which means you're throwing away 80 to 95% of your polynucleotide, which is an expensive commodity. But this new tech caused spontaneous assembly of these positively charged lipids and the negatively charged polynucleotide. And they were mixed with other lipids that were fusogenic. They would cause fusion with cell membranes. And if you formulated these in the right ratio, they would stick efficiently to the surface of cells, cultured cells, and pull the polynucleotide in, and you would get, in the case of what Syntex had been able to do, DNA transfection. So you could get DNA without a virus. Remember, I'm in a gene therapy lab focused on viruses for getting polynucleotides in, and get that DNA into the cell, eventually into the nucleus, and make protein. So Tony suggested I contact Gordon. Gordon put me in touch with a guy named Phil Felgner, who eventually became my boss at Vical and was a collaborator then for a few months once this had happened. And I exchanged a series of emails and also filed a patent disclosure for the Salk before I contacted Phil, having to do with using RNA as a drug. And Phil sent me this new reagent, and suddenly everything worked like crazy. And it was like you couldn't fail. The reagent was really working well. And what was a little ironic is that the Syntex crew had tried to make RNA delivery work, and they'd failed, which is why they were so glad to work with me, because they hadn't done all this other stuff that I'd done before to build the platform.
Right.
So it was one of these moments in time where things came together, and suddenly I was able to efficiently deliver RNA into every single cell type I tested. And I went around the Salk Institute, gathered all of the different cell lines that everybody had, including caterpillar cells, because of people were starting to work with baculovirus. That's the platform that the Novavax product uses. So this was, back in the 80s when all this technology was just kind of starting to happen. And I gathered all these, tested them, and then I did a series of experiments to show that, with statistics this time and linear projections and R-squared values and all that kind of stuff, to show that you ' a linear dose response with the RNA, and compare these different structural elements in the RNA to show that the cap was important, and the 5' untranslated region was important, the 3' translation, and the polyadenylation was important, et cetera, et cetera. So that led to this PNAS paper. That is really the foundational paper, and everybody still uses that basic construct and the same enzymes and basic technology that I scaled up to milligram levels, only now they do gram and kilogram synthesis of RNA based on those same enzymes. So that's that part of the platform. In parallel, I had a postdoc that was kind of shepherding me, named Dan St. Louis, mentoring me. And Dan was working on a retroviral gene therapy project in which he would use the retrovirus to infect cells in culture, mouse cells, and then create a little artificial implant of those cells and place them into a mouse and observe the expression of the foreign transgene, a potentially therapeutic protein, in the mouse. And this method worked quite well for about 3 weeks, and then it just shut down, and there was no more protein expression. And everybody was scratching their heads because I mentioned that this was a gene expression laboratory, and so they were thinking about elegant ways for gene silencing in retroviruses integrated into chromosomes. And I pored over the textbooks from my first 2 years of medical school. And I came up with the hypothesis that, no, it wasn't any of that fancy stuff. It was that the mice were generating an immune response against the foreign transgene. And this was heresy. This because what it meant was the whole logic of gene therapy was a failure.
Right.
Because the fundamentals of immune response, and this was not an immunology lab, right? So that's and it meant that the truth is that if I have cystic fibrosis or muscular dystrophy or whatever, so I have a bad inherited error of metabolism in my genetics, in my chromosomes, and I'm producing a defective protein. It's defective in the terms of it's associated with my disease. My immune system doesn't know that it's a defective protein. It knows itself. It knows that bad protein is actually the normal protein for my body. That's the way the immune system works. And so if one introduces the good gene that doesn't have those genetic defects, the thinking was, this is how we're going to cure pediatric disease, inborn errors of metabolism. That was Ted's, because Ted is a pediatrician. But instead, what you run into is that the body will reject the good gene because all it knows is not that it's good, but that it's different.
Right.
And so suddenly, here I am in this pressure cooker gene therapy lab, having decided I was going to commit my entire life to retroviral gene therapy, and suddenly it's clear that ain't going to work.
Right.
And we, Houston, we got a problem. And so the brainstorm was, okay, so let's make lemonade out of lemons. We can use the gene therapy technologies for vaccine purposes. We can turn the whole thing on its head and make what's suddenly a disqualifying disadvantage for gene therapy into a major advantage for a whole new technology space, a whole new way to approach vaccinology. So now I'm going to circle back. I mentioned Dinko Valerio, the top status PhD in the whole laboratory that went on to create Crucell. After we both left the lab, I saw him at a gene therapy conference about 3 years later. And he took me aside and he said, Robert, he had already started Crucell at the time, which was focused on gene therapy. And he said, Robert, you're right. I'm going to pivot Crucell from being a gene therapy company to being a vaccine company. And that's what then Crucell exploded, was sold for, I think, $1.5 billion or something, and to J&J. And that is the lineage that gives the J&J vaccine. So they both, the mRNA vaccines and the adenoviral vectored vaccines, trace back to Inder's lab in the late 80s with this Dan St. Louis. And Dan didn't even make up much of it. He didn't really talk about it much in the paper. And I am included in the acknowledgments as, Bob Malone. We thank Bob Malone for his contributions or something like that. But meanwhile, back at the ranch, I had, as a graduate student, you have to make your nickel somehow. I had a National Science Foundation fellowship, but one had to do teaching assistant rotations. And I was assigned to work as a teaching assistant on an embryology lab. So one of the tasks that I had to do was to prepare frog embryos at different stages of development from African clawed frogs. This is one of the core technologies used in modern embryology and developmental biology, is the model system of Xenophus laevis. So I was preparing these eggs and fertilizing the eggs and making the tadpoles and whatever. And at one of the preparations, I had tadpoles at a certain stage of development, all timed. And I had way more than I needed for the class. And so I thought, everything else works with this. Let's try frogs. And lo and behold, it did work. So I took the same RNA, the same RNA reagents, and then also did this with a DNA that gives a colored reaction, beta-galactosidase. Put them on the frog embryos, and lo and behold, we got luciferase, and then we got beta-galactosidase. The beta-gal was localized to the eyes, and that led to other things that other people did subsequently in embryology. But then the next step in the embryology course was we had to prepare something called chick in a cup, which is a way to prepare chick embryos so that you can observe their development. And so again, I had extra chicks that I prepared from fertilized eggs. And in that case, took the same method, reagents with DNA, put them on the chick embryos, and lo and behold, that worked. So now I'm sitting on dynamite in terms of intellectual property.
Right.
And I've filed a patent disclosure with the Salk Institute that they determined I was the sole inventor for, having to do with the use of RNA as a drug and RNA delivery into cells in animals. And that's when the problem started. And it became such a storm, dropping the first part of that word, that I got caught in the middle of a huge fight over who would own this. UC San Diego, the investigator who'd been the professor for the embryology course, thought she should be an inventor. Inder thought that he should be an inventor because he was my mentor, not because he contributed.
Right.
The president of the Salk Institute, De Hoffman, called Inder on the carpet for setting up this illicit collaboration with Syntex, because at the time they were trying to route all the intellectual property into their for-profit subsidiary of the Salk called Salk Institute Biologic Associates. And so Inder got called on the carpet. I got called in and asked, "What the heck have you done, young man?" And I told the story, and Inder flat out denied knowing anything about what I'd done with the Salk Institute. I'm sorry, with Syntex. Tony later told me, "Well, what did you expect? Of course he lied." The cognitive dissonance got to the point where I had a nervous breakdown.
Really? Is that bad?
And I was diagnosed as having PTSD by a UC San Diego physician. And, the crossfire of the attorneys at UC San Diego telling me I couldn't talk to the people at the Salk, people at the Salk saying I couldn't talk to UCSD. I was just a pawn. I mean, literally, Mongo is a pawn in the game of life. I was Mongo at that point, if you remember Blazing Saddles as the reference. And it was overwhelming.
Wow.
And I had to leave the laboratory. The laboratory was an abusive environment anyhow, and your listeners can look up Inder Verma in Science Magazine and find out things about Inder that we don't want to go into right now. So it was a very abusive work environment, but I just came unglued, and I still kind of have the scars a little bit from that.
Sorry about that.
That's the way things are. It happens to a lot of graduate students. It happens to a lot of graduate students. That's part of what gives me the comfort in telling this story and being open about it, is I've had many former graduate students, including from the Salk and Scripps, that were in this pressure cooker environment where everybody thought they were going to get rich. I mean, that's what's really driving this. We talked about that. It's always all about the Benjamins.
That's right.
And people smelled money, and they wanted it, right?
Yep.
And it ended up, I didn't get any compensation for any of this, except reading forward one Susan B. Anthony dollar from Vical for the rights to all my patents, the stuff that I contributed. So I needed to get out of the lab. My wife needed to finish her bachelor's, and so I needed a job for the next 6 months or so while she finished her bachelor's degree at UC San Diego. And the guy that I had been collaborating with, Phil Felgner, and that I'd put on the RNA PNAS paper over Inder's objections, had been recruited as the first employee of a company that had been set up by Carl Hostetler and Doug Richman called Vical. And Vical was set up in the old cyclotron that was right across Torrey Pines Road in the other direction from UC San Diego, which was small lease space for biotech startups and engineering startups. So the old cyclotron was pretty grubby space. And Carl had created this company together with Doug Richman to look at antiviral compounds for Burroughs Wellcome that were liposomally captured. So this was the early days of AIDS antiviral development, and Doug Richman was right at the forefront. And so he had a contract to take anti-HIV antivirals and put them in liposomes so they'd go into macrophage, which was the reservoir for the AIDS virus. And Carl wanted to make calcitonin analogs, Carl Hostetler. Carl went on to create Triangle Pharmaceuticals with Ray Shinazi, and that's the hepatitis B antivirals, and great things happened.
Sure.
Carl's had an illustrious history. But that was the genesis of Vical. It was founded, I think, in '88. Phil joined in late '89, and he said, "Robert, since you got to leave Inder's lab, why don't you come work for me? And we will give you the money to set you up with a little skunk works in the corner and set up your molecular biology." And so I brought over my reagents, my plasmids, actually some RNA I had made, a bunch of this lipofectin reagent that Phil had shipped me from Syntex. So I actually brought the lipids. I was a mule, I guess, for transferring these drugs from Syntex into Vical by way of the Salk. I was naive and just trying to get my science keep moving.
Sure.
And they set up a collaboration with a guy named John Wolfe, who had just left. He had gotten burned out on retroviral vectors. I'd known him when he was at Ted Friedman's lab. And he'd been burned out on retroviral vector gene transfer and got a position as a pediatrician at University of Wisconsin. And UW gave him some access to some animals, rats initially, in animal facilities. And so Phil convinced John to collaborate on this new technology that had, come out of Syntex and out of Inder's lab. And so John was going to be the animal injection characterization group. I was the, manufacturing the polynucleotides and kind of the core technology. And then we had a couple chemists at Vical that were working on the other projects, the liposomal nucleotides, and they started synthesizing novel cationic lipids that would be outside of the patent of Syntex. And all this came together with me shipping John samples, including blinded samples, of mixtures of initially DNA plus cationic lipids. And John didn't believe in luciferase yet. That's how new it was. And that it would be useful in vivo. Eventually I converted him. And the initial cat assays were positive. And I think the initial studies in rats were done with DNA, but then we tested the RNA in mice. And I have the first data is posted on the website of those. Those have never been published, those first experiments. But I have the initial chloramphenicol acetyltransferase films from the first mRNA experiments. And this is another part of this amazing story. Initially, the lipid RNA complexes worked in the mouse to everyone's amazement and joy. It was like, hallelujah, here we are. And one of the chemists named Raj Kumar was a great skeptic and said, you guys aren't doing good science. Now, I had done the negative controls, I don't know how many times with the frogs and the chicks, and it never worked. The negative controls being not including the cationic lipid with the polynucleotide. But Raj insisted that we had to do this with the mice also, and that and basically to shut Raj up, to be honest, just to make him go away and stop yapping about negative controls that were irrelevant. I shipped John the samples with and without the cationic lipids and cationic lipids alone. So I did the full spectrum of positive and negative controls. We already knew that the cationic lipids plus the RNA would work. So that was the positive control. And his postdoc injected them, worked up the cat assays, and we got a fax back that showed that, yes, once again, the cationic lipid plus RNA had worked. And I said, we get on the phone. This is old school technology,
right?
Old school technology, came across by fax. And I got on the phone and I called up John, landline, of course, and no, hardly any,
Right.
We had like intranet for, and we thought that was pretty cool. And I called up John and I said, yeah, but John, this lane over here is positive, and this was supposed to be the negative control. And he says, the technician must have screwed up, and some of the assay from the adjacent lane must have been in that one. And I said, okay, we got to do it again.
Right.
And so I sent John more samples, and lo and behold, the RNA alone was as good or better than the RNA plus the lipid. And then we did it with DNA, and that led to the science paper that's so famous that came out in 1990 that came out just a couple of months after the cold fusion paper came out. And the world reacted that this was another science screw up, and cold fusion applied to molecular biology. But in this case, everybody all across the world could reproduce it. And so the patents were written about how this, and I spent hours and hours in downtown San Diego with a top-notch patent attorney talking about how this could be used in all the various applications. And I actually disclosed the use of targeted mutagenesis to produce what we would now call gain-of-function mutations in influenza that could be used for vaccine purposes to build a universal influenza vaccine. I don't think that Vical ever completely comprehended that patent disclosure, but it was way, way ahead of its time, like by 2 decades, 3 decades,
right,
as was most of this stuff. So I because of my time with Bob Lamb and my time with Murray and Preston Marks and all that, I had a pretty good handle on influenza vaccinology and HIV vaccinology and the use of antisense. And I was communicating with Tom Scheck about this is before he got the Nobel for ribozymes, about the use of ribozymes to stabilize the RNA. And just my mind, was on hyperdrive then. I was right at the forefront of all this. And my supervisor really felt uncomfortable, I think. I think a good supervisor, it's good to try to hire people that are your match or perhaps even better intellectually, and manage those people. This guy was uncomfortable with that. It made him very uncomfortable. And he started really aggressively trying to claim credit for what I had done and what I was doing and signing off on invention disclosures. And it was just clear that I couldn't stay as a technician, with a master's degree. I had to go finish my MD. Jill finished her bachelor's, and I resigned from Vical. And I was only there for about 4 or 5 months. And all of this technology came out of there. And it was all orthogonal to the Vical business plan. And so they hired some people. Stan Gromkowski is one, Gary Rhodes is another, Suzanne Parker is another. with assistance of Doug Richman, they set up the influenza model in mice to try to verify the things that I had invented, disclosed, conceived of, etc., using an influenza model, and it worked. And they were, with Stan's help, they were able to demonstrate not only did it generate antibodies, but it also generated cytotoxic T lymphocytes, which was what the theory was, that this would more replicate a natural viral infection, so you get a more complete immune response than with the standard vaccine. And so the patents ended up issuing. Vical sold all these patents to Merck for a little over $9 million, which at the time was a lot of money for them. Absolutely. They were gasping for air at this point, and for capital. And so this $9 million would fuel their other programs. And the whole thing was just not in the business model of Vical from the outset.
Right.
Merck this was Merck under Maurice Hilleman, Merck Vaccines, before Maurice Hilleman died. So the brilliant vaccinologist that created many of our pediatric vaccines in the mid-century era. So Maurice was still there, and Merck insisted that they be able to take credit publicly for the inventions and the work. And so Merck basically took what Doug Richman and the team had done with the mouse model, repackaged it, put their own people on the paper in senior positions, and got it published in Science, and that just set the world on fire. And everybody was off on a race to build DNA vaccines, which is there had been a decision when I left. I think nobody could really make the RNA. And so Vical decided, well, we're going to go with the easy thing, which is the DNA. And that was the fork in the road. Merck also looked at the RNA and decided that just didn't make sense chemically because RNA stability and the manufacturing cost, etc. And they were going to only focus on the DNA. They spent many years, never developed a product, spent gobs of money, and gave up, returned the technology to Vical. Vical then spent tons of investor money, well over $1 billion, never could make a product, and eventually went bankrupt when the patents expired. But what they did in the interim was they were very aggressive legally, both Merck and Vical, and they kept everybody else from doing anything with these ideas and the technology, including me, sending me as a then young academic cease and desist letters saying you will not work on these things that you did when you were with us previously.
Wow. So
that's a story of how science really is, not how it gets into the textbooks as all happy, and we're all seeking wisdom and enlightenment. That's modern biotechnology is really driven by profit, and it distorts a lot of things. So you asked, and that's the most complete discussion I've ever given on how this all came about, and thank you for giving me the opportunity to share it.
If you don't mind, Robert, I'd like to get into the meat of now the current vaccination for COVID, if that's okay.
Yep. Yeah.
I think the first question that I have is in terms of the FDA approval timeline. And I understand, and I think it's important that we make it clear that you are not involved at a government level currently, or even with any of the big pharma companies, correct?
So not involved with the government is a little more nuanced. Let's take that one first. Please. Well, no, the pharma. The pharma is dead easy. I have no financial historic or present consulting relationship with Pfizer, BioNTech, Moderna, or CureVac.
Okay. So
those are the lead companies. I do have a long-standing professional relationship with the gentleman, the senior emeritus professor at University of British Columbia, Peter Cullis, who is the one that, in my mind, has made the most important enabling subsequent discovery, which is the development of an improved cationic lipid, different from the quaternary amine. It's ionizable amine that has enabled the amazing in vivo, so in humans and in animals, delivery efficiency that we see, and is the basis. They actually license his formulation technology and his composition of matter for both CureVac and BioNTech Pfizer products. In a case of, I assert that the Moderna product technology is reverse engineered of what Peter and his group have developed over the last 40 years. So, so there's that. But I have that's just a professional relationship like I would have with any other academic.
Sure. And
I have no financial conflicts of interest relating to these vaccines. On the government side, I've had very active interactions with some senior members of the FDA that reside outside of the review branch. So FDA is parsed into different segments, and you generally can't interact with the review branch people that are actively reviewing INDs or other regulatory documents associated with licensure or authorization to proceed with any of these vaccines. So there's that. With one should know that I got a call from Wuhan from a person that's part of our intelligence community who was in Wuhan during the fourth quarter of 2019. And he called me the first week in January, I think January 3rd, thereabouts, and said, Robert, you got to get your team spun up for this new virus. This is a big one, and it's looking like it's going to be important and a major threat agent. At the time, I was a senior member of a team working for Department of Defense on a contract for screening and developing agents, drug agents, as countermeasures for organophosphates, which is to say chemical warfare agents and also pesticides. So I had a team that included people in high throughput screening, in structure-based drug design, and in computational analysis, high throughput analysis of existing compounds as potential inhibitor agents, Josh Patel. And so I convinced this team, well, the sequence was published either January 10th or January 11th as the Wuhan seafood market virus. And I got that sequence, immediately used some computational tools to convert one of the key protein sequences, the papain-like protease, into using the known structure of the papain-like protease from SARS, and converted that to a predicted crystal structure for SARS-CoV-2 for that protein. And then we started docking, which is this computational process of fitting molecules into a known binding pocket. It started docking all currently licensed compounds as a drug repurposing effort. I had done a threat assessment at that time, and it was my assessment that there was no way that we would have, with the standard licensing process for vaccines, no way that we would be able to have a vaccine in a timely fashion to mitigate the risk of this disease. And the best way, knowing what I know about regulatory affairs, to come up with something that would help people was to repurpose existing drugs. I had previously created a company called Etheric, working closely with the Department of Defense and USAMRIID to screen repurposed drugs for Zika. So I knew this space. That company had gone bankrupt because there was no capital interest in repurposing drugs, because you can't make a lot of money on them because they're already off patent. So I knew the space, and that's what I focused on, was drug repurposing, and have since. So this whole issue of the vaccine toxicology is something that has come to me. I've been aware of it and cautioning my colleagues at FDA about certain aspects because I know the core technology and know what it can do and what it is doing. And there were things that they really weren't thinking about that I tried to help them think about. So that's how I got involved in this vaccine toxicity story.
Okay.
And then there was a series of other events that involving physicians, discussions, and that I'm also rigorously trained in bioethics, as is my wife. And actually previously had been a whistleblower in the Jesse Gelsinger death case with the adenoviral vectors. So I've been through the scrum, but usually with the press, I don't seek attention. I stay on background. But the issues here were such, particularly the bioethics, that I kind of came out. My primary customer, DoD people, they don't really like me to be talking in public very much. And they're tolerating this, but they're biting, they're gritting their teeth a little bit. So that's, I don't have the COI. I do work closely with the government. I've been working on, over this span on 5 different government contracts. I've captured over $130 million in government contracts specifically for a drug, among other things, drug repurposing and platform trials with a company called Quantum Leap Healthcare out of UCSF. And we currently, I'm a subcontractor to Leidos, a very large government service provider, and we're moving forward with an IND, so getting ready for the large double-blind randomized clinical trials for the combination that I've developed together with some investigators at Beloit Memorial in Wisconsin. That is the drug combination celecoxib and famotidine. This has solid mechanism of action based logic as opposed to ivermectin and some of the others, and really strongly positive results. So those trials are about to start. So that's my COI here, if I have one, is that I've been deeply embedded in drug repurposing, but been brought into this discussion about the vaccine toxicology, which I know quite a bit about also, and regulatory aspects and the bioethics. As a kind of an outside observer with deep knowledge and the ability to explain core concepts and issues to folks that aren't necessarily deep in the gears of dealing with this stuff. Does that help? Yes. Have I answered your question?
You have. And in terms of the drug repurposing, I want to make it clear for listeners. What are you exactly saying? If you can explain drug repurposing for this case, if you can explain that so people are very clear on what you're talking about.
Oh, sure. And there's a video clip from back in my etheric days. I think it's 2017. The time passes. 2017, and you can find it on Contagion Live on their podcasts. Me talking about drug repurposing. If you want to get deeper into it.
Okay.
The core logic is that we have a large pharmacopoeia, is the technical term, of drugs that have been licensed over time for various purposes. And so the way it works with the FDA is that one obtains approval to market. That's what licensing really is, and interstate commerce. Technically, that's why the FDA has the authority to do this. It's interstate commerce to market and provide agents which have therapeutic or prophylactic benefit that have met all the safety criteria and are manufactured in a way that is consistent and not adulterated and potent, etc. And so one obtains these authorizations for a specific purpose, but physicians are allowed by their licenses to use any of the agents in the pharmacopoeia in general, within certain limits, to treat their patients. So you can take a chemotherapeutic drug and you may use it to treat a autoimmune disease or a chronic inflammatory disease.
Okay.
And it may not be labeled for that purpose, but as a physician, you're allowed to prescribe it for that purpose. So those are the rules. And as a physician, you have to operate within certain boundaries. You can't just do crazy stuff because otherwise you'll get sued. So you have to kind of stay within the bounds of standard of care. That's the drug landscape in general. There are, because we now have so many decades of experience, and as I mentioned before, patents are only good for 20 years in general. Drugs that have been marketed for a particular purpose, a great example is ivermectin. The investigator at Merck that developed ivermectin received the Nobel Prize for his discovery over 40 years ago. So ivermectin is off patent, as one example. Hydroxychloroquine is another one that's off patent. So what that means is that generic drug manufacturers can come in and produce those drugs and make them available to the public at greatly reduced rates. And we all know about that. We go to Walmart and the pharmacist says, yeah, I'm going to give you the generic, right? And why are they going to give you the generic? Because your insurance will only pay for the generic. They don't want to pay full price for the patented one.
Right.
So that's how that part of the drug industry works. But these agents have many activities, and they may have activities that have not yet been discovered, which is why one uses these kinds of tools like I was describing, of computational docking and high throughput screening and high content imaging. And there's a whole bunch of cool new stuff that is very AI, cloud, etc. driven,
right?
That involves big data, that allows us to set up assays with, you've seen them in all the movies, the little plates with all the little wells. So these are high throughput assays that involve machine screening, etc. Wife and I were just watching the Tomorrow War, which is just up, I think, on Netflix, and that kind of tech plays a big role in the storyline. So you can see examples of that kind of a modern approach in that, Hollywood piece. So high throughput screening, computational sequencing, artificial intelligence, deep learning applications, a lot of cool stuff. And what's done is you can apply, we have libraries of compound structures now that are billions of compounds. And you can write a purchase order for high density plates that are all plated with different drugs and different drug candidates, and then run them through your assay and see whether or not they inhibit your virus replication, your protein, and what have you. That's the cutting edge right now is that world. And so with repurposed drugs, you can take those libraries of things that are already licensed off patent, and nutraceuticals, and all these exist as compound structure libraries and compound libraries that you can just write a purchase order and get them shipped to you, and as high density plates. And so the logic is that one screens all of these drugs to find the few hits, that's the language, that look promising for some new indication. And it may be related to the known mechanism of action, or it may be a new mechanism of action for the drug. And many, of course, I'm not the only one that does this and knows of this kind of approach. And so many labs all over the world started doing this. And we were one of them, maybe one of the early ones, but many followed. And so that's the logic, is that one identifies a compound that is ideally off patent, generic, which means inexpensive, typically widely available. The safety profile of that in humans is already known because it's had to go through the licensure process. The chemistry, manufacturing, and controls dossier to produce that at safe and unadulterated, as a safe and unadulterated product, is already known.
Right.
And so that cuts out about 2 to 3 years. You know, you have the toxicology is already known, the manufacturing is already known, it's already being produced at scale by various generic drug manufacturers. The safety in humans is already known. All you have left to do is prove efficacy or effectiveness for your new indication.
Right.
That shaves a ton of time off of the life cycle, which normally is a decade or more and $1 billion or more.
Wow.
Okay, so that's the core logic of drug repurposing in the teeth of an outbreak. Is all of this stuff, the RNA vaccine technology, the DNA vaccines, and all the stuff I've done for decades, is all wrapped around how can we rapidly respond to either an engineered or emerging pathogen. I stopped being an academic and started working for the government right after 9/11, and worked for the DoD under contract on virtually all of the vaccines and biodefense products at that time: smallpox, anthrax, V, etc. It goes on and on. Tularemia. So that's the way things are in that kind of world that I live in now, with Defense Threat Reduction Agency, Joint Science Technology Office. The domain program kind of partially comes out of my head, which is wrapped around how do we use these kinds of ideas like drug repurposing to rapidly respond to an outbreak. So there's the logic of drug repurposing. It's a potential pathway to rapidly providing pharmaceutical agents as opposed to prophylactic vaccine agents that would be able to fill in the gap between when a novel biologic threat enters the population, either because of deforestation or whatever other thing,
Sure.
Bats or whatever, the Ebola being an example, or the bad guys have done garage biology and engineered the darn thing, which is increasingly the big risk. Let me just close on one last fun little fact about this, since you have
Please.
a wonky audience. One of the key truths here is that the Department of Defense is on track to have vaccines for all of the biowarfare agents deployed through the end of World War II, historically. Is tularemia and plague and smallpox and all that. We'll have vaccines for all those by, if everything stays on schedule, which is a problem, by 2050. Okay, so a full century after all of those were deployed. Okay, so clearly that is not working. We got to come up with something better, and that is what drives current thinking in the Department of Defense and the people that I work with, who are supporting the warfighter, not the civilian population.
Right.
And that, and how do we respond to engineered threats, particularly in terms of protecting the special forces that are small strike teams that go into threat arenas, and so that's the world that I currently work in and have since 2001 for the most part.
Robert, and I want to make it clear that you're not saying that drug repurposing would replace a vaccine. It is literally to mitigate the risk currently. That's what it would be for, to take masses and mitigate risk down with millions of people infected with whether it's COVID or another virus, correct?
Correct. And it has, so they're not competitive.
Right.
They're complementary, right?
Right.
And the logic was back then that if we had the ability to obtain drugs that were active in reducing, and knowing that they would not be perfect, right?
Mm-hmm.
And knowing, remember, I come from the AIDS world. So the thing that beat AIDS was not a vaccine, as we know, unfortunately,
Right.
but rather the David Ho largely gets the credit for the brainstorm of using multiple drugs that constrain the ability of the AIDS virus to evolve in your body.
Right.
And this has to do with, it's a big numbers game. If you have a 1 in 10 to the 6th probability of developing an escape mutant against one drug during your infection, and a 1 in 10 to the 6th probability of escape mutant from another drug, and likewise for a third drug, as you know, as a technical person, your probability of escaping all three is a product. Right. And so we get down to the teeny tiny numbers, which is what you have to do with viruses because they have huge numbers of progeny. And those progeny have mutations, and that's what drives viral evolution. It's a very special, extreme case of evolution. Viral evolution is. And we're dealing with that right now with the escape mutants, by the way. So the logic is with the drug repurposing in antivirals is to come up with multi-drugs that each are imperfect, but act together in a way that is complementary and idealistic. You know, in the best of all possible worlds, they're synergistic, but that's often difficult to get to. But even if they're additive in terms of their activity, you can really get something much more powerful out of things that are imperfect. While the great brains of Pfizer and Gilead and Merck and everybody else are busy developing new stuff and working along that 10-year trajectory, God bless them, we kind of need something to take care of folks now. And so that's the logic here, and it's absolutely not, it's complementary. Even with, as we know, with Delta variant, right? These miraculous vaccines that we have, and what's happened with the technology, both the adenoviral vector technology and the mRNA, both genetic vaccines based on gene therapy, the results have exceeded expectations, gently put, but they're not perfect either. They're not blocking viral infection, replication, or transmission. And so we still need agents, even in a perfect world where everybody takes vaccine. Right. We're still going to have viral infection. We're still going to have disease. And anybody that doesn't believe that, all they have to do is look at the Israeli data or the British data right now with the Delta variant in highly vaccinated populations. So absolutely not competitive, complementary.
Okay. Do you believe in the vaccines currently offered?
Belief, I try, as a scientist, I really try to stay away from belief. I try to stay on data. And what we have is a situation where, like with every vaccine, there is no perfectly safe drug. People like to say even water taken at sufficient doses is toxic. And just to make the point as you take a drink. And the same is true with vaccines. There's no perfectly safe vaccine. The question is always with any product, what is the relative risk associated with the product when you take it, and what is the nature of those risks, and what is the relative risk of the thing that you're trying to mitigate or treat. And in the case of this disease, COVID, which is different, COVID disease the hyperinflammatory reaction of your body against the virus. Okay, it's different from there's a viral syndrome that happens soon after you get infected that is truly more like an influenza. But there are characteristics of this virus that provoke the immune system of some people to just go nutso. And that is a subset of people that get infected that develop that. And that, it turns out, is inflammatory in nature. And the good news, the reason to underscore that point, is that it's wicked hard to develop effective antivirals, things that directly block the virus. And with respiratory viruses, historically, like flu, Tamiflu, we all know Tamiflu, right? Donald Rumsfeld made lots of money in this company called Gilead. It's really hard, especially with respiratory viruses, to come up with an agent that will really impact the disease course. And to the extent that they do impact the disease course, even if they're very effective in cell culture, they have to be administered very early on at a time in this virus when most people don't even know they're infected. So that's the history of, and in the case of flu with remdesivir, with Tamiflu, you have to take it kind of within the first 24 to 48 hours of being infected, which is really problematic. Right. I've got a runny nose. I feel kind of crummy. I'm going to go to the doctor and he's going to give me a drug for flu. Yeah, good luck scheduling your flu appointment unless you go to a doc in the box. It's just that it's not practical. And that the same is true with COVID. But the good news is that we do have a rich library of drug agents for treating inflammatory diseases, because they're profitable, right? So we've got all these things, and a bunch of them are off patent, and it looks like a number of them have activity, and they're not perfect, like I said. So there we go. Proper coughing technique demonstrated. So that's that space. I hope I've answered whatever that question was that I was reacting to.
Do you think that there's a certain vaccine, Moderna, Pfizer, that a specific person should take? And I know it's hard because you can't categorize every person and everybody's.
Good. So this follows the thread that I was just developing, but I got lost on. So thanks for bringing me back on the reservation. In general, you won the right and proper way to proceed in this area. Once you have enough data to make evidence-based medical decisions,
right?
Is that which we didn't have at first, so people had to substitute expert opinion, but now we can make data-based decisions. And what you do is, and have always done, what the CDC has always done, is to evaluate what we call the risk-benefit ratio. I was referring to that earlier. The risk of disease from the pathogen in this case, which is distributed across the entire population, versus the risk of associated with the intervention vaccine. And you stratify those risks into the various key populations. And the way they're usually defined is infants, children, 2 and above, adolescents, healthy normal adults greater than 18 and typically up to 65, elderly, 65 and above, pregnant women, and special populations like immunocompromised or people with allergies or other pre-existing risk conditions.
Okay.
And you do the risk-benefit ratio for each of those populations, and then you make a decision to recommend you should take vaccine if you're at this risk-benefit ratio. But if you're not at that risk-benefit ratio, you don't take vaccine because you might cause more harm than good. In this case, we have a disease. I went over that a moment ago.
Sure.
The disease, COVID-19, is primarily disease of the elderly, and your risk goes exponential the older you get, and the obese, and people with certain pre-existing conditions, which we don't completely understand yet. Vascular leak syndrome is one that's very clear. Other types of inflammatory diseases, potentially cancer and other conditions, certain types of cancer being immunosuppressed from chemotherapy, those kind of things. So the answer to your question is, it's complicated because and there are algorithms, by the way, that are AI ML based. So it's, as a techie, you'll appreciate that.
Yeah.
That just do an awesome job of predicting your risk of developing severe disease or death. And the AIs have taken into account some of the key variables are your zip code, the size of your house and the density of people in your house, indirect measures having to do with socioeconomic status. And these are AIs built off of non-HIPAA controlled data. So they're not actually built off of healthcare data that's protected, but all of the amazing data sets that all big tech has on all of us, Amazon, etc., that you can buy these databases, run the AIs through, and they come up with quite remarkably predictive scores.
Interesting.
Now, if I was king, I would say, let's make those available to everybody so they can figure out their own risk and make their own decisions about vaccination. But in their infinite wisdom, the government has not decided to take that path at this point in time. But it could be possible, and you can inform yourself about your risks. It's not easy. Your risks of disease, a lot of that you would have to be somebody like me that's comfortable diving into the primary literature.
Sure.
Or you could rely on your doctor, who's probably going to say, just take the jab and shut up. Because they don't have any time, because they're not budgeted for doing that. They're budgeted to give you a jab in the shoulder and tell you to move on. And to really make your own personal assessment of risk-benefit ratio, you have to understand what the risks are. And again, in their wisdom, the government has decided on the noble lie strategy of denying that there are risks, or only disclosing risks if they're absolutely forced to, and just insisting that everybody get the jab, and putting out a whole huge army of deploying amazingly powerful tools to convince the population that they should take the jab. But those old gray hairs like me that have been through this and through multiple outbreaks say, yeah, but if the data show that if you're an infant through about the age of 40, this is increasingly clear even with the limited data we have in understanding of the risks of these genetic vaccines, the adenovirus vectors plus the RNAs. The risks for newborns through about age 40 are at the margins or upside down for taking vaccine versus your risk of getting COVID, severe COVID, to the point where you're hospitalized or you die. So many of us are saying, no, this has got to stop. We can't, especially these are still experimental products. And so this gets into the bioethics, the fundamental bioethics that go back to the Nuremberg trials and the Helsinki Accords and the Belmont Report here in the States, and it's codified in federal law as the common rule, all say that for an experimental product, there must be full and complete disclosure of risks. Those risks have to be comprehended and comprehensible, so that in the States that translates to 8th grade language.
Right.
And the person has to accept the experimental product of their own accord by free choice, free will. They cannot be coerced, and they cannot be compelled through incentives like lottery tickets or shotguns or whatever
Right.
else, or ice cream for the adolescents. And in the case of adolescents, and infants and children, we define the age of consent as 18, and so by definition, they cannot provide informed consent, and their parents or their guardians have to. So those are the rules, and we have decided to. The government has decided to disregard those rules, even though they're in federal law, and put out all this information and censorship to try to keep people from even discussing things, even scientists discussing risks or other aspects of all this. So don't put this up on YouTube, you may get deleted. So in terms of the specific vaccines, in my opinion, I've been asked this question countless times, as you might imagine.
Sure.
And I have deep knowledge of the adenoviral vectored technology. The thing about all of this is that the FDA did not apply the gene therapy regulatory checklist to these products. They only applied the vaccine checklist. And so they didn't require that the developers do genotoxicity or full reproductive toxicity, and they didn't require them to characterize how much of the protein insert in the gene therapy technology. In all cases, this is the viral spike protein with 2 amino acid modifications that were designed to make it more immunogenic, not designed to make it more safe. So that's a misconception that Reuters has promoted. They were not required to characterize where this protein was being expressed, for how long, in what cell types, and at what level. So none of that is known.
Is that frightening to you?
It's concerning, frightening. I took Moderna twice. I probably shouldn't have. I did have adverse events from that on top of my long COVID. I developed grade 3 hypertension, restless leg syndrome, and other symptoms that I hadn't had before.
Interesting.
So the hypertension is one of the adverse events that's associated in some people. And I'm not the 135-pound young buck that I once was anymore.
Right.
So I have to blame myself for not being in perfect shape also. But just knocking off the adenoviral vectors in the States, that's the J&J product. Adenoviral vectors were designed for long expression of high levels of protein for gene therapy purposes. And so I infer, in the absence of data, because they weren't required to characterize this, that there will be substantial amounts of protein transgene, spike protein, produced from the adenoviral vectors for long periods of time with those vaccines. I have suggested generally that people avoid those vaccines for that reason.
Okay.
Now, talking about the RNA ones, there's only 2 on the license, or they're not licensed, I apologize. These are all under emergency use authorization, so they remain experimental. Yep. Of the 2 mRNA vaccines, one has Pfizer and Moderna. Remember that Moderna is a company that was created by DARPA, and the vaccine itself was engineered by the Vaccine Research Center at NIH. So it's good to know that Dr. Fauci's National Institute of Allergy and Infectious Disease receives substantial royalty revenue from Moderna, as do 6 different staffers who have government salaries. They get supplemented $150,000 a year for royalties from their work that they did in engineering what we now call the Moderna vaccine.
Wow.
The Pfizer vaccine is based on BioNTech, which was similarly sponsored by the German government. Okay, so those, it's good to know that those are government-sponsored companies.
Right.
And they're government-backed with the PR push and everything else. So it's just helpful. In terms of the Operation Warp Speed events that happened, OWS, Pfizer, Moderna kept their products basically outside OWS. They were competitors. And the mRNA vaccine that was the focus of OWS is the Moderna product. The Moderna product dosing in terms of RNA is about 3 times that of Pfizer, 3 times higher.
Wow.
And when you look at the adverse event profile from the VAERS database as shown by CDC, some of their publications, the Moderna product is associated with higher and broader adverse events than the Pfizer product. And furthermore, the adverse events are more prominent with both of them with the second dose than with the first dose. So based on that, in general, if you have the option, I suggest that Pfizer is probably the way to go here in the States until Novavax becomes available, should it become available, because there was a decision apparently in HHS that these 3 vaccines were sufficient and we didn't need a more traditional vaccine like the Novavax product that's made in caterpillar cells using an adjuvant derived from tree bark. So Novavax pivoted to an agreement with CEPI to make this product more available on the international market. And I'm not even sure if it's going to be made available in the States. One hopes so, because there's about, over 50% of the population right now are vaccine hesitant. And I think many of those would prefer a more traditional product than a genetic vaccine product. But I don't know if the government's going to make that an opportunity for us. Many countries do have more traditional vaccines. Often they're less effective for preventing death and disease, although now we're seeing that the high effectiveness levels of the currently licensed products, if we believe the Israeli data, is not as strong against some of these other variants like Delta than they were against the original Wuhan strains. So there's inside baseball about how OWS came up with the Moderna dosing, but basically it was decision making by committee driven to consensus. It wasn't really so much science-based. And there's some recent work out that shows that you can get really good immune responses with the Moderna product at about 1/4 the dose with only a single dose, not 2. So people ask me what next, or government officials occasionally say, well, what can we do? And my response is we can really look hard at dropping the number of doses and dropping the overall dose of certainly the Moderna product. But that's all speculative. It's just, like I said, inside baseball communications. And for folks like you in your listenership, we're kind of stuck with what the government allows us to have access to.
My next question, Robert, is it's an interesting one because millions, hundreds of millions of people have received a dose, right? So the question becomes, yes, it's an experimental vaccination or vaccine. The question is, how can the governments, and I say governments because it's a worldwide issue, not just a North American issue, right?
Absolutely.
The governments turn around and say, well, we've decided we're not going to pass this we're not going to approve this vaccination because it doesn't meet our requirement after hundreds of millions of people have been vaccinated.
You're dead on. Politically, they can't do it.
Right.
And this gets back to the consequences of what I call the noble lie. And the logic, what we've really had happen here, by the way, this emergency use authorization strategy where we really don't collect data on the people in terms of the safety during this phase, and we rely on this really kludgy, antique database system involving self-reports where we can't, here in the States, because of HIPAA, we can't couple people's medical records with the report that exists in VAERS to be able to really get a rigorous hunt on whether or not those are just temporal associations or are they functional associations. And just to illustrate, with the Ebola vaccine, what the government did is they used something called expanded access or expanded use access. And what that meant was that they created these massive clinical trial structures. And anybody that needed the vaccine would be enrolled in one of these open-ended clinical trials. And they would be treated as if they were in a standard clinical trial environment where their adverse events would be captured both through solicitation. That's, ring, ring, call center. How you doing? Did you have this, that, or the other thing?
Right.
And self-reporting or unsolicited adverse events. And the government, in their wisdom, decided not to do this, and they were hoping that they could kind of cobble together a bunch of different databases from the military and other things. That hasn't worked out so well, and anybody that works with healthcare databases knows why, because they all have their own little nuances in coding. This is a tech problem of harmonizing different databases, which most that are involved in computer technology understand exactly how painful that is. And so doing this in a rigorous way has turned out to be overwhelming to the CDC, which is not staffed by armies of Atlanta computer scientists, because they're all making 10 times the budget that CDC would have, right? Working for big tech. So that hasn't worked out so well. And so they're in a position where worldwide now, these different databases are uncovering safety signals. It seems almost daily, and having to wrestle with what I call the noble lie, which is embedded, by the way. Someone recently on Twitter pulled out the Federal Register from 1984, an auspicious year, in which the government clearly lays out that largely for the polio vaccine campaign, any information about that raises concerns about the safety of that vaccine was to be suppressed, actively suppressed, circa 1984. And that has long been a public policy, as I mentioned, the noble lie, the idea that leaders and key public decision makers, public health leaders and government officials are justified in lying to the public for the common good. And I haven't read Plato, but I've learned a lot over the last few months about these things. And this traces back to the work of Plato. So it's sometimes called the noble Platonic lie, and you can wiki it and find more than you ever wanted to know. But when you see the communication, the emails disclosed in the Washington Post of senior government officials that I'll not name, openly admitting that they've been lying to the public, for the public good, this is grounded in a Platonic concept that it's okay to do that for the common good, because people wouldn't accept vaccine should they actually know the full spectrum of risks.
Right.
And so the logic is that anybody that discloses those risks or discusses those risks is causing people to die because they haven't accepted vaccine. And so that gets into this latest Dirty Dozen stuff about a list of people that have been identified by the government as causing death because they are discussing vaccine safety risks. So and this is happening worldwide. There are countries where physicians are being declared mentally incompetent for either raising these questions or for advancing or discussing repurposed drugs or alternative therapies for outpatients, for instance. And some of these physicians are actually being institutionalized for their mental instability for having done so. And they're definitely losing their licenses or their ability to practice. So that's what's happened, I think, here, if you'll forgive my venturing into this space.
Please. No, please.
We have this idea of enforcing the noble lie that goes back, clearly at least to 84, if not earlier, in public health. So this is almost a mid-century idea in American public health and worldwide. It's been carried forward through time without really being examined logically. Does this really make sense? Is this a good idea in the modern age where everybody wants to Google everything and find out anything they can? And if the pieces don't fit, or they feel like they are being manipulated and the information is being manipulated, then they go into what I call Lego conspiracy mode. There's all these little fragments of information like Legos that can be assembled to your favorite conspiracy. And that is actively happening because this is how people's minds go. Intelligent, thoughtful people are prone to, if they're not given the straight information, they're prone to fabricate stories of their own based on the little bit of information that they have. And that is actively happening, and it's driving the government nuts. I think that this idea of the noble lie and restricting access to information and disclosure is antiquated. I think that's really what's going on. Is we've had this intersection of horizontally integrated capabilities for information control that are amazingly powerful, that didn't exist even during the Ebola outbreak.
Sure.
So the trusted, if you want to look up Trusted News Initiative, it's not a conspiracy. It's actually a BBC press announcement, British Broadcasting Corporation. And there's this organization of major news media and tech like Facebook and Microsoft, etc., Google, that are integrated and were built as a system, an integrated system and consortium for purposes of resisting foreign disinformation campaigns during elections. And with the rollout of the vaccines last fall, there was a decision that this organization would be turned and used for this purpose of the noble lie of suppressing any information that was contrary to the established narrative. So that's why your YouTube will be taken down if you talk about things like ivermectin or vaccine safety that that involve concepts or discussion about information that is not consistent with the official World Health Organization party line or the CDC. And yet we all know, because we've lived it, that the CDC has flip-flopped multiple times, the WHO has flip-flopped multiple times, and other public health leaders that will be unnamed have flip-flopped multiple times and are actually quite open about it. They think that they've done it for a good reason. But that's, you get big tech and in the media all integrated. And now we have funding coming from Bill. This is all true. It's not a conspiracy.
Absolutely.
We have money coming from Bill and Melinda Gates and the Wellcome Trust that is flowing down and creating new pop-up fact-checking organizations that are really applying what we would traditionally call psychological operations concepts to craft public perception about all these issues and motivate the public to accept vaccine across all age cohorts.
Right.
And that's I think that what we really have, there's a lot of people that go down all kinds of rabbit holes about Davos and collusion and the grand scheme and the big reset and all this stuff.
Yeah.
And it's just about a vaccine. That, I can't say that isn't true. I can't, I don't know the data. They don't invite me to Davos. I haven't sat on those meetings. I don't know that actually happened.
Right.
But I can see the structures, because it's all public, for this information campaign that is integrated. And we have it's very open and transparent. We have interlinks between big pharma like Pfizer and the press. So the chairman of the board of Reuters sits on the board of directors of Pfizer. I mean, it's all integrated now.
It's crazy.
And it's incredibly powerful for information control.
Absolutely.
And it's often, you know, any of us that work in tech at all understand algorithms. And, you know, I like to say artificial intelligence is the least intelligent of the various options there, right, down to deep learning, right? Artificial intelligence is like the big hammer. It just hits things. And it's kind of stupid in a lot of ways. And so that approach, and that's what we're dealing with, is algorithm-based fact-checking, where you're comparing statements that may be nuanced with broad brushstroke statements that have been approved in the language approved by the World Health Organization or the CDC or the European Medicines Agency or fill in the blank National Health Authority. And there's other subtleties to this that are fascinating, if you don't mind my going there.
Absolutely, please.
Both of our sons are computer scientists.
Okay.
And fairly high end, even though they're young, and they make more money than I do. And one of them, who works for a company based out of Atlanta that does, you know, distributes billions, literally daily, emails, and has all kinds of cool tools that I won't name.
Please.
But he had this conversation with us the other day about scattering cockroaches. And it's that metaphor is not very, you know, it's not politically correct speech to use the metaphor of scattering cockroaches, but the concept applied to the Facebook decision to delete the user groups that had been established to discuss adverse events that people claim to have experienced about vaccines. Okay, so they deleted 150,000 people or something like that were all chatting about their experiences because they thought this was going to dissuade people from taking vaccine. When they did that, they essentially stomped on the cockroach nest or on the food that attracted them, is the metaphor. And the cockroaches all scattered into other venues, Reddit or whatever, you know, Instagram,
sure,
blah, blah, blah. They were no longer aggregated in a single place where the government could have data mined that quite successfully,
right,
to provide signals that they could then apply statistically to the CDC databases, because that's the core problem with analyzing these databases is multiple imputation statistically. You can't ask at a 95% confidence interval the broad question, is there any association of any of this stuff with vaccine? Because you get into a statistical trap when you do that. And you end up just statistically with 5% of the stuff, if you have a 95% confidence interval, being false positives. And you end up chasing the false positives. And it's wicked hard to get down to the true positives if you take that type of approach. So statistically, what you have to do is you have to have a hypothesis. Oh, I think that this is associated with cardiac events. And then you go looking for testing the hypothesis. And then you run into other problems like masking and confounding, because if you have a high baseline of, say, cardiac events in the older population, you'll never see the new events that trickle in from vaccines. But if you have a low incidence of cardiac events, say, in adolescents, you can see that signal quite clearly. Okay, so it doesn't mean that it's not happening in everybody else. It just means you can't find it statistically. So that's the kind of, now that we have this situation where we are sampling the worst possible type of data, self-reported, that cannot be associated or not associated, that it's previously, years ago, documented by a Harvard study that the VAERS database, this type of self-reporting, results in an undercounting of about 100-fold. So when you see Steve Hirsch with whatever hair he has left, with his hair on fire, about the number of deaths in the VAERS database, which cannot be fully attributed or ruled out as vaccine associated because of the structural problems. What's really setting Steve's remaining hairs on fire is that, I'm teasing Steve Kirsch in case he ever listens to this, is that that's probably a 100-fold underreporting of the deaths. Okay, based on previously peer-reviewed published research.
Sure.
So we're in, and as time goes by, and what's happened is that the governments worldwide have used these profoundly powerful tools of information control and population control in terms of knowledge and bias to impose a bias on not just those that they wish to take vaccine, that there's no problems here. That same messaging and bias runs all the way through the CDC. And the CDC has it as a mission that they should comport with this overall logic that adverse events should not be openly disclosed, which is in direct conflict with federal rule and the common rule and the Nuremberg Code and all that stuff. But they believe they're doing it for the right reason. And that gets us to this weird situation we're in right now. And what's starting to happen is that enough people are getting red pilled that they're starting all over the world. Intellectuals in particular are starting to say, this doesn't look right.
Yep.
And now the government, as you say, with your question, has got a problem. And they can't lose legitimacy.
Right.
And yet, a fun fact, it used to be that 1% of the population had self-identified as anti-vaxxers, and now it's over 40. And, I put out twice, once on Twitter and once on LinkedIn, a comment, what happens to trust in public health if it turns out that ivermectin has efficacy, not perfectly effective, and is safe, and vaccines are not perfectly safe, which is the messaging we received. And both those went viral. LinkedIn viral means 20,000 people. Twitter, not millions. But with just comment after comment after comment, we've already lost our faith in public health, in the WHO, the CDC. As I've said in other podcasts, what I'm concerned about is what happens after. And this basically, these people that had been labeled, gaslighted, pejoratively labeled as anti-vaxxers, it's a common slander, now are being, their historic reservations about government integrity and the behavior of the pharmaceutical industries are being validated.
Right.
And I have people writing me and commenting all the time, I don't want my kids to take any vaccines now.
Right.
And so in this, I think that we don't have to go to the Davos controlling the world conspiracies. I think that the Occam's razor here is what we have is incompetence and groupthink, compounded by the acceptance of this historic logic of the noble lie and mapping it into a new technology and media space where the tools for controlling information that are now perceived as increasingly authoritarian are amazingly powerful.
Absolutely.
And people are waking up and seeing that, maybe for the first time, and saying, "Ooh, this doesn't feel right at all."
Agreed.
And in the politically, we didn't want to go political. What I find fascinating is that this is orthogonal to the typical, the classic left-right axis.
Yeah.
It is, the good news is it's allowing, what I'm experiencing, it's allowing people that traditionally have been in opposition under this left-right axis to be interacting.
Right.
And establishing what they believe is the world that they want to live in. And most of them are saying, no, we heard George Orwell loud and clear, and we don't want to go there. We don't want a ministry of truth, and we don't want newspeak, which is another way of saying politically correct if you think about it. And we're not okay with I think the real where the rubber hits the road is this logic of forced vaccination for infants through young adults and young adults returning to university. That isn't supported by the risk-benefit data. That was a long monologue. Thank you for letting me get it out.
No, thank you. Do you believe that the government will, or the governments will, kick the can down the road, so to speak, and approve the vaccine because they really don't have another choice? And on top of that, with companies making millions of these vaccines per day, I presume, right, in production, at what point do they look at the vaccine and go, okay, we have to modify this component and change? It might be the same ingredient, if you will, in layman's terms, but the milliliters of it are just reduced, right?
Right. So dose adjustment.
Right.
Or reformulation.
Right. The reformulation mainly, like in terms of, because the governments are in a really bad position right now, as we've talked about now for quite a bit of time, and they're really not going to have much of an option to just FDA approve this and we move on and everything's good, but they can reformulate.
So I have to be careful here because I have spoken with senior government officials on the Hill about this problem. And, and you're correct that I believe strongly we need to approach it with empathy and not as setting up yet another conflict, even though that's what the media loves, is to set up a binary conflict. Divide. Yeah. So how does the government get out of this hole? Is the part of that question. And will they kick the can? The poor soul who is right at the pressure point on this is named Peter Marks. And he's the head of the Center for Biologics Evaluation and Research at the FDA. And the rest of the world really does look to the FDA still as the premier regulatory agency. Unfortunately, the FDA has been very significantly compromised by regulatory capture from the very industry it regulates. This often happens in American politics. USDA is a notable example. So Peter, bless his soul, is right at the fulcrum, and I'm sure he's under enormous pressure to approve, because it would reduce the pain of the government. The government, I think, has cognitive dissonance, just like many of us do.
Right.
And cognitive dissonance is pain. So how are they going to reduce their pain? I think, and I've suggested to senior government leaders that they have the option to resort back to standard protocol for the Advisory Committee on Immunization Practices. And rather than pushing the logic of universal vaccination, they roll out recommendations that are stratified by risk. So that's the first thing. And that I think a lot of people worldwide, and there are many court cases now prosecuting this. So this, stay tuned. This is going to get a little rough and ugly. Buckle your seatbelt. There are there's a logic that neither emergency use authorization nor licensure market authorization should be granted for the pediatric applications, and probably also should be excluded pregnancy, and maybe some others.
Wow. And
that would allow the government to take a modified limited hangout, referencing back. So you get it. So
yep.
I think there are some modified limited hangouts here for the government that will allow them to save face and still maintain access to vaccine. I was on a podcast for 2 hours yesterday morning, starting at 7 with an international group, and they wanted us all to agree to a consensus statement that the vaccine distribution should be halted at the moment. And I said, I can't sign off on that. The risk-benefit ratio for the elderly is there. And furthermore, if you do that, I didn't say it, but it is, you're tilting at windmills. This is not going to get you anywhere. You got to focus, in my opinion, on the things that you can win.
Right.
And I'm focused on trying to protect infants and through young adults. I think that's a winnable thing, and I think it is solidly supported by data. And I think the government could get behind that and say, you know what, we've kind of rethought this, and we look back at our policies, and the data have changed, and then there would be articles out in the Washington Post, editorials saying, I think this is a great idea, and in the New York Times and the Wall Street Journal, and we would all say, and everybody would turn around and say, sounds good, off we go.
Right.
March. I think that they really should make Novavax available in North America for those vaccine hesitant that are just uncomfortable with the genetic vaccine, which is like about half the population, and give them another option. And it's sitting there waiting for you. And if it means that you have to scale up production for Novavax, but even just saying that they will make Novavax available under EUA would take some of that political pressure off.
Sure.
And many people believe that there will be a decision to license the Pfizer product at least certainly by Christmas, if not sooner. You asked that question.
Mm-hmm.
And I tend to concur that is a likely outcome. And but what's happening is that folks with deep pockets are aggravated enough about this that you can buy, for instance, the whole Medicaid, Medicare data package for the last 2 years for about $25,000 and get access to all that data and do your own data analysis. And I know of people that are doing this, and they seem to be discovering that the CDC is grossly underreporting many of these adverse events. Just based on the CMMS data. And that's when that comes out, and it will, the government is going to have a real problem, and this is going to end up in the Supremes.
Really?
I can't see how it doesn't. This is, if the government stays entrenched like this, they're risking major political blowback and legal blowback that it will tear apart the current public health system, and really already is. And I think we've got key constituencies politically in angry mothers, young adults. And by the way, the African-American uptake of these vaccines is about 20% because they have this historic memory of Tuskegee. And Tuskegee is a CDC thing. And they really don't trust the government. You think that the Ruby Ridge types don't. African-Americans as a bloc are pretty dug in that they don't like a lot of things the government does in public health. So those are three key constituencies that, if alienated, will transform the political landscape, gently put. And so where does this go? And you had these enormous protests against Macron in France with his mandate. The vaccine passport logic is a trigger. People just are going nuts over that.
Absolutely.
And it's like the government. That's why I say it's groupthink. I think worldwide, a leadership has convinced themselves they want to come to consensus. They want to think alike. They want to be aligned. And I think they have gone down a rabbit hole, and it may be in part because they've been influenced by these mega donors that we don't need to name. We mentioned them earlier. And I think those mega donors have built up their own logic and are true believers in the vaccine strategy rather than kind of pragmatic, which is how I try to approach it, of kind of open-ended.
Sure.
What makes sense here? What makes sense there? And let's pick a little from this, Chinese menu approach.
Yep.
They're all in on vaccines is the way to solve the world's problems.
Right.
And they've spent a ton of money influencing the World Health Organization and governments to get with the program.
In terms of alternative, I don't want to use the word medicine, but treatment. Are you in agreement that vitamin D and zinc daily is a great way to combat this as well?
Combat is a strong word.
Okay.
I'm more in the reduce your risk camp.
Okay, fair enough.
And absolutely.
Okay.
And likewise, I think that my colleagues, my valued colleagues who believe so strongly in ivermectin as a solution, and some that I've been on a podcast with even believe that ivermectin can eradicate COVID and the virus from the population. They're not epidemiologists and virologists, and they aren't outbreak specialists, and they don't know. There's a lot of internet experts out there. The practicalities of doing something like this, and the necessity of having an agent or a vaccine that is incredibly potent, that can knock virus replication down to extremely low levels, like we've done with HIV.
Mm-hmm.
Getting to the point where we had drug therapies that could control HIV and make it so that those that are infected have almost a non-existent risk of transmitting the virus to a third party has taken 2 decades. Ivermectin ain't it. It has activity, both as a prophylactic and a therapeutic. I'm convinced of that. Does that mean that it's the silver bullet? I'm convinced that it absolutely doesn't mean that. And does it mean that it is a extremely potent antiviral at safe doses? Absolutely not. So that's, but the vitamin D and zinc story, there's widespread vitamin D deficiency,
right,
including myself. And I'm having to take fairly large doses. My cardiologist is actively managing me as an elderly, young gentleman. And that's part of the supplements that I take. And, we can measure my vitamin D levels, and they're not where they need to be, and they're getting there.
Right.
So that's just good medicine. Taking care of our bodies, reducing our body weight, staying fit, and getting plenty of sunshine, and making sure that the measurable variables that are associated with immune competence are where they should be. That's just good medicine, in my opinion.
Dr. Malone, is there anything that I haven't asked you that I should have, and that you would like to say? This is your time.
Okay, so the concluding statement. I mentioned the noble lie. The noble lie has 3 elements to it. The first is that the way that we get out of this economic situation we're in, because, of course, like I said, it's all about the money. The way that we get out of this economic situation and reduce disease and death morbidity and mortality for COVID. The thesis is that the only way to do that is to reach herd immunity. The second part of the noble lie is that the only way to reach herd immunity is with vaccination. The third part of the noble lie is that vaccination is perfectly safe. All of those are false. Okay, so there's, drugs can help us with the disease and death.
Sure.
And the way that we usually get to herd immunity is through viral spread of the natural infection. So there are those that call this natural immunity. And the vaccines are not perfectly safe. We know that. How safe or unsafe they are, we're still debating that.
Right.
That's the first one. Second one, key point, is that the bioethics here foundation has three components. So 3 and 3, easy to remember. There has to be full and complete disclosure of risks. Those risks have to be comprehended, and you have to accept those risks if you are to accept, particularly an experimental product, of your own free will. You should not be coerced through public media management or anything else, or free shotguns, to take vaccine. You should take it because you think it's the right thing for you, which puts the burden of responsibility on your shoulders, by the way. This is kind of a libertarian piece, this common rule in bioethics. It's kind of grounded in the idea that your body is yours. It doesn't belong to the state. Right. And so with that comes responsibility. And if you wish to abdicate that responsibility and not think about these things and just go along to get along, that's fine. That's your choice with your body. And if there are downstream consequences, you will have to deal with them. And the liability protections are such that you can pretty much forget about getting any compensation if anything happens to you. So that's just the way things are. The third is a good news piece. The history of virology is that as a virus enters a new population, a new species, it tends to be highly pathogenic very often. And that is the case here. Whether or not this was engineered in a laboratory, it crossed over from bats, it's kind of irrelevant. It's in the population in terms of this. And what happens is that over time, the evolutionary pressures on viruses in a population drive them towards becoming less pathogenic and more highly infectious. If you think about it from a virus's point of view, you don't want your host to go to bed or die. You want your host walking about, talking to people and shedding virus and infecting other people.
Right.
So those are the evolutionary pressures, and I suspect that we will get there. That this virus is embedded and will be embedded in our population for the rest of our natural lives, for better, for worse. But it will gradually become more like a flu or a common cold. That's the hope. And that's the history of other viruses. The last point I'd like to close on is the Rodney King plea. Can't we all get along? If we go with what the media likes, media likes these binary oppositional relationships. It really sells papers. It's super good for clicks, great for advertising to say, you're either anti-science or you're pro-science. You're either pro-vaccine or you're anti-vaccine. It misses the gray, all of this information in between, but it serves their interests because they're not scientists. They're not public health officials, and they can talk about politics and give their opinion about the fight. They're horrible at understanding the nuances of what's going on, but it's really easy for them to do the play-by-play of the baseball game and what inning we're in and who's winning. And they love to do that because it's something they can do, and everybody can understand that. They don't have to spend time on their segment like we're spending 2 hours just to get down to the basics. And don't play into that. What I like to say is we need to share with each other and have an open heart and appreciate, as you do, that the public health officials and your practicing daily physician who has spent all of his or her life, professional life, being taught to just do what the CDC tells you to do and don't think about it too much. And you're asking that person to say, process the cognitive dissonance of this crazy guy Malone and others like him, Dr. McCullough and others, the Dirty Dozen, saying, maybe there's something going on here and we ought to pay attention, and maybe, we're no longer in Kansas and everything is perfect. And we need to live in the real world. Well, that doesn't kind of fit with what they've been taught to go along with whatever the officialdom is telling them.
Right.
And so they're having psychological pain and cognitive dissonance. The public health officials are in a bind, as you point out. Mm-hmm. And if we set it up as a fight, it will continue to be a fight.
Correct.
And if we approach it with open hearts and open minds as a shared dialogue, then I think we can move towards solutions that are suboptimal, not perfect,
sure,
but more adaptive than what we currently have. So that's what I'd like to close on, if you don't mind.
Thank you so much for taking all of the time. I really do appreciate it. It's fascinating to learn about a new billion-dollar industry that none of us expected would come about. And I thank you for your work, past and certainly present, and look forward to hearing more from you.
Thanks for your time and for this forum.
Thank you so much, Doctor.
Thanks for listening. Find us on YouTube and Facebook at the Intellectual People Podcast and online at theintellectualpeoplepodcast.com. Check back for exciting new episodes.